It'd be pretty sweet to learn about sugars, right? So let's do it. But let's not just focus on sucrose, like table sugar. Nah, let's talk about all the cool sugars we can get, starting with glucose, so our simplest sugar that our body uses. Building up, yeah, we'll talk about the sucrose, but then we have things like our glycoconjugates and our proteoglycans and our proteolipids and all this cool stuff. So much more to talk about with sugar than just what you think about on your table.
When we think about just like sugar, we're probably thinking of sucrose, which is what we call a disaccharide. It has these two individual sugar units linked together. These individual sugar units are called monosaccharides. These include things like glucose, our blood sugar. These can then link up to form all sorts of different things that can serve structural roles, such as the starch in plants, or storage of energy, such as glycogen that we store the glucose, all that glucose in our bodies in this form of glycogen that we can then like chew off when we want energy. And that's just part of it. There's also, if you attach those sugars to other things like lipids, so like the fatty membranes around your cells, or proteins, or like peptides, so short protein pieces, there are basically tons and tons of different things that sugars can do in your body. And in various organisms, they can play different roles. So you might have heard of like endotoxins. So bacteria actually have these glycolipids, this LPS, where they have these sugars. These bacteria have these distinct sugars that they put onto the surface of their cells. And then this actually, it protects them, but it also causes us to have an immune response against them. And this can cause some of the symptoms from a bacterial infection. That's just one example. So that is an example of a glycolipid. You'll see that there's a lot of different terms for describing different glycoconjugates. So a glycoconjugate is when you have some sugar part, so the glyco part, is actually attached or conjugated to another molecule, such as a protein, a peptide, or a lipid. And we'll get much more into these various things. But I just wanted to orient you to there are some different terms that we use to describe these different conjugates. And I was always really, really confused about these because we have terms like glycoprotein, proteoglycan, and peptidoglycan. So proteins are made up of chains of amino acids. And if you have a long chain, we call it a protein. And if you have a short chain, we call it a peptide, a really short chain of peptide. And so when we talk about peptidoglycan, that's going to be the sugars that are kind of connected through these peptides. And bacteria use this to build their wall, and we'll get much more into this. But okay, so that makes sense. So peptidoglycan, you've got the peptides and you've got the sugar. And now we come to glycoproteins and proteoglycans. Well, what's what? So the key is that the second word is going to play the starring role, and the first one is just going to be like modifying it or that sort of thing. So in a glycoprotein, the protein is the main part, and then the glyco is this smaller part. So you have mostly protein and then some sugars attached. And actually, like I think like half of proteins have at least one sugar, and then some proteins have a lot of sugars. These sugars can do really important things. We'll get into some of them. They can also cause problems. And so, for example, like viruses and things they often have glycoproteins on their surface, such as the coronavirus spike protein. It's a glycoprotein. It's got a bunch of glycans sticking out. These sugars are then making it harder for the immune system to actually recognize. So that's a glycoprotein. It's mostly protein, and then it's got some sugar chains attached. When we come to proteoglycans, now here we have the sugars are going to play the starring role. So it's mostly sugar, and then we've got some protein in it. And these can serve as important scaffolding hubs, as well as they can attract water and form gels. So when you think about mucus, when you think about fluid in your joints, these sort of things like lubricating, these things are often proteoglycans. And so we'll get much more into these conjugates. I just wanted to start off with some of these terminology. But in order to really appreciate the combinations of sugar with other things, we first need to focus on the sugar itself. So we often use this term sugar to refer to like carbohydrates, and carbohydrates are going to have this general formula, which is CH2O. And so basically what this tells you is that you have one water molecule per carbon. And so that's a lot of water molecules. And in order to fit all these water molecules in, they're in the form of this hydroxyl group or alcohol. So sugars are very, very alcohol-y. They have a lot of these OH groups, which might seem kind of boring, especially if you compare them to like amino acids where you have all of this variability. So they're all very varied in terms of what functional groups, so what different like sorts of reactive groups that they have. But when we come to sugars, and then we're just like, okay, well, we have hydroxyls, we have some carbonyls, so this is where this is oxidized to this carbonyl. And this is oxidation is one of the modifications that can happen to an OH group. And there are other modifications as well that allow you to have a lot of different variability just having those simple groups. So even if we're talking about monosaccharides, so where you have a single unit, such as, so this would be like glucose, galactose, fructose. So you can see that there's several different ones. These are just a few of them. There are actually 9 main monosaccharides in our bodies. And these can also get modified as well. And these can link up in various ways to give you various molecules. There's some terminology around glycobiology that can get kind of confusing. One thing that is meant to simplify but can sometimes be really confusing if you don't know what you're looking at is that we can often abbreviate these various monosaccharides with colored shapes. So you'll see that glucose, it has this abbreviation GLC, and it's represented as this blue circle. The shape of it is going to tell you kind of something about its structure or its modifications. So the circle, it signifies that these are hexoses. So what a hexose is that means it has six carbons. So the number of carbons is going to dictate what kind of sugar is based on this nomenclature. So if you have 3, it's a triose, 4, it's a tetrose, 5, it's a pentose, 6, it's a hexose, etc., etc., etc.
We can classify sugars as aldoses or ketoses, depending on whether they have an aldehyde, where the carbonyl is at the end, or a ketose or a ketone, where the carbonyl is in between 2 other carbons. Now, there's a position. So where we see this aldehyde or ketone, basically that's our anomeric carbon. And so it's anomeric because what's going to happen is that another, like one of the hydroxyl groups lower down on the sugar chain can actually go and attack it. And it attacks that anomeric carbon, and this is going to ringify the sugar. And it's going to basically, depending on whether it attacks from the bottom or from the top, you're going to have the part that's kind of like after it be sticking up or sticking down. And so if it's sticking up, we call it beta. So this would be beta. And if it's sticking down, we call it alpha. And these are different anomers. So this is the anomeric carbon. It gets attacked, we get different anomers. And depending on which kind of hydroxyl the attack is coming from, you're going to get different numbers of sugars in your chain. And so like the part that was down here is now up here, and the part that was over here is now down here. And so if you had a ketose, you would kind of have more sticking out because your anomeric carbon was sandwiched in between 2 other carbons. You can interconvert between alpha and beta, but to do that, you need to break open the chain, but then you can do it. So we see here, we have this alpha, and then if we see this one, is going to be beta. So beta is sticking up, alpha it's sticking down. So when we draw them with like, this is a Hawthorne projection. And when you see it kind of in a straight line, that's going to be your Fisher projections when we're talking about showing the stereochemistry. But these are all examples of Hawthorne projections where we kind of have the flat ring and then we have things above and below the ring.
But when we talk about like this D, this can get kind of confusing because it has to do with stereochemistry, typically, but this D and the L is actually referring to how the sugars like last OH groups are oriented in space relative to the simplest monosaccharide, D-glyceraldehyde. Okay, so basically it has to do with how things are oriented, not the beta alpha orientation, but more in terms of the actual like stereochemistry as a linkage. So differences that you can't just interconvert between. So there's this idea or this concept of stereochemistry where things are like kind of like things can be kind of like your right and left hand, where they look like you should be able to just like convert between them and overlay them. They look like they're the same thing, but you can't. If you try to overlay them, they're non-interconvertible. And so this is the case with like this D and L. It's easiest to see this when you're looking at sugars in like this linear form. And then, so then this is being compared to the glyceraldehyde. So don't confuse this with like the technical definitions of DNL typically used when we're talking about stereochemistry, in that they represent the rotation of light. So different molecules with different stereochemistries can rotate light in different ways. So if you have two molecules and they differ at one location based on the stereochemistry, so like which way things are facing in like in space, then they're going to rotate plane polarized light differently. And this is one of the ways that you can tell them apart. And so depending on which direction they rotate things, so dextro or levo, then you call it D or L. So that's how the terminology is officially used. With sugars, that all goes out the window. It's all just compared to that one sugar and compared to glyceraldehyde. So you have some D sugars that are actually really, they truly are rotatory, so they're rotating light to the right, including D-glucose. And so we got lucky that they named the glucose the D because it is dextrorotatory. And so doctors often sometimes call glucose dextrose, which I always get really confused about. But yeah, so glucose and dextrose, same thing. But there are also other D sugars that are actually levorotatory, so don't let that confuse you when you're dealing with that. And so D-glucose is the form that our bodies are like using. Okay, so the modifications, as I said, that OH group, that hydroxyl group, it might seem really boring, but it can do all sorts of really cool things. It can get aminated and like acylated and stuff. And so when you have that, this is called, so you get GLC, NAC. So this is N-acetyl D-glucosamine. So amine, it's got this amine, this nitrogen group in there, and it's got this acetylation. And this is going to make this an amino sugar. And we're going to see these come into play with some of our conjugates that we talk about. It can also get oxidized. And so oxidation is the loss of electrons or electron density. It's often accompanied by the gain of a bond to oxygen or the loss of a bond to hydrogen. Much more than you need to know right now, but you can get this oxidized to this carboxylate state or this carboxylic acid if there was an H here. It can also be reduced, so you can like remove that oxygen and now you have just a hydrogen. And so this would be like a deoxy sugar. You might have heard of like deoxy in the context of DNA. That D is for deoxy. And this oxidation, just a fun note, so this is glucuronic acid or gluconurate. This carboxylate group can go between like carboxylic acid and carboxylate just by gaining or losing a proton, this like H plus. And so you'll see this written as either gluconurate if it's in this deprotonated state, or gluconuric acid if it's in this protonated state. And you'll often just like see it one or the other, even if it's, I mean, because you don't really know what it is exactly. You can guess based on the pH what proportion is going to be on what state, but those are interconvertible. So you'll see it written both ways. Okay, then we can so we have all these long names and stuff, which can get kind of confusing. Since we have those main 9 ones, we can then represent them with these different shapes and colors. The square represents an N-acetyl hexosamine. So hexose, remember that 6 amine it's got the amino group, and then it's got the acetyl group as well. So this N-acetyl hexosamine because it's got 6. Okay, then the acidic sugars are going to get diamonds. So remember we talked about how you can get oxidized to see this carboxylate groups. And this one too, just another note that N-acetyluramic acid is also known as acetyl sialic acid or sialic acid. Various modified forms of these are also called sialic acid, so that can get kind of confusing as well. And then you have weirdos that get weird shapes. So deoxyhexoses get triangles and pentoses get stars. So pentose would be five sugars. Okay, so in an example is xylose. Okay, so now let's connect them together, because one of the cool things that OH groups can do is they can actually like attack one another. They can link together in different ways, and different sugar monosaccharides or different individual sugar units can link up together. So you get different combinations of sugar units and different ways that they actually link together. And so when you have two of them linked together, we call it a disaccharide, and they're going to have different properties based on what sugars they contain, what monosaccharides they contain, and how they're linked up. So a couple common sugar disaccharides are sucrose. So this is just like table sugar. It's got a glucose and a fructose attached in this alpha 1,2 beta linkage. So don't worry about the nomenclature, but remember that alpha or beta is basically whether this group is sticking up or down, because when it ringifies, that last group can stick up or down. And so based on which it is, then that's going to dictate what kind of link, how you call the linkage. And it's also going to dictate like kind of like the orientation. When we get into like complex carbohydrates, this is going to matter a lot how they link, because it's going to kind of be like trying to put like what shape the ultimate carbohydrate is going to make, which is important implications in terms of its structure, and sturdiness and tightness and compactness and all that stuff. Another example is lactose. So here you have a glucose and a galactose attached through this beta 1,4 linkage. People who are lactose intolerant don't express an enzyme, so this reaction helper that is needed to actually cut these apart. Another example is maltose, and here you have two glucoses, and they're attached to this alpha 1,4 linkage. Okay, so those are disaccharides where you had two, and later we'll talk about complex carbohydrates where you have a lot. What if you have just like shorted chain, more than two, but not a ton? We call it an oligosaccharide, and an example is raffinose, which is the carb in broccoli. So we can't digest it, but the bacteria in our guts can, and they let off gas as a byproduct. So think raffinose for that. Okay, so now let's get complex carbohydrates. Typically, these are used for structure or energy storage. So if you want structure, think about what you'd want in terms of these various things. So if you have structure, you want the strands, you have a lot of strands of these sugars, you want to kind of like keep them close together. So you would want straight chains. An example is going to be cellulose. So this is the plant wall sturdifier. It's made up of glucose, and it's attached in this way that it's going to be linear. And so this is going to allow lots of points of contact between the two strands. Lots of contact, but what if you want to make things even sturdier? So with the sugars, remember, we just have these hydroxyl groups, and these can form like hydrogen bonds with one another and stuff, but they're not great. I mean, they can be really strong, but what if you need it stronger? Like you wouldn't want to have an exoskeleton, so you wouldn't want to have like a shell made up of cellulose. That wouldn't be sturdy enough. You need something sturdier. And so like crustaceans have this complex carbohydrate called chitin. I can never pronounce it right. But it has modifications that are going to offer up additional hydrogen bonding opportunities. But these modifications will make this sturdier and stiffer, so it makes a good shell. We'll get much more into this later too, but it's not always stiffness that you want. What if you want something more jelly? And so sugars can form really nice gels as we take advantage of in the lab, such as with an agarose gel electrophoresis, where we can get sugars to form this mesh filled with water, so a gel. And then we can use that mesh to separate DNA pieces by size. And so this comes in really handy, and it's taking advantage of the fact that all of those sugar groups can attract water, especially, as we'll see later, if you have modifications on them, such as negative charge sulfate groups. This is going to allow them to really soak up a bunch of water and form a gel. So, and yeah, depending on the amount of modification, you can get different sturdinesses. Okay, so we don't have cell walls, we don't have shells, but we still need energy. So we still use complex carbohydrates for energy storage, and we use it in a form called glycogen. Plants use a storage form, the starch, amylopectin, and basically, when you have a energy storage form, you're going to want it to be really branched because this is going to allow you to have a lot of points to start chewing from. So if you only have a couple ends, well, then you only have a couple spots to start chewing off from. But if you have a lot of branches, well, now there's a lot of places where you can get to work kind of chewing down in order to get energy really quickly from it. So when you compare like the starch amylopectin and human glycogen, they both have alpha 1,4 glucose chains and alpha 1,6 branch points, but glycogen has more branching. And what's really cool is it's actually anchored around a protein called glycogenin.
Glucose is really great. We can break it down for energy, and it's a main way in which we store energy in our bodies. But if we were to store it as glucose, well, that would be a big problem because of osmosis. So if you had a bunch of individual sugar units, thinking back to like colligative properties and all that jazz, what would happen was that all those individual sugars would kind of dilute out the water in a way. And so the water would move into the cells thanks to osmosis. And what would happen was that the cells would burst. And we don't want cells to burst. Well, at least not in our bodies. Sometimes we do in the lab, but we don't want that to happen here. And so what instead we do is we take these glucose and we hook them up into glycogen. And so glycogen has this huge, big branching structure. It has these long chains with these branches, and at its heart is this protein glycogenin. And glycogenin, well, it is actually an enzyme. And so glycogenin is going to take that first role in putting together the first pieces, and then other additional enzymes will help out. So for like the straight chains or alpha 1,4 linkages, so we are hooking up the 1 position to the 4 position to give us this alpha 1,4 and it's alpha because of the position where this oxygen is sticking down rather than up. This is an alpha linkage. And so this is an alpha 1,4 linkage, and it's giving us this straight chain. When we looked at glycogen structure, we saw that there were a bunch of branches, and to add those, there's another branching enzyme. There's a branching enzyme, and the branching enzyme is going to take a part of the chain and kind of shift it over, so we get this alpha 1,6 linkage, where we have the branching from the 6th point. So in this way, our body is able to store glucose for when we need it. And so when our bodies, they signal, your pancreas sends out insulin, and then cells get the message to take in glucose, and the liver and muscle cells take it in, and they link it up into glycogen. But they don't just like they're storing it for a reason. And so for your muscles, they're like, I'm going to want energy later, and I don't want to be out of energy, so I'm going to store some for fast use. And then when I need it, I will break it down. So that's a process called glycogenolysis. And then I will use that. Basically, I'll get glucose that I can use to make ATP. That's not technically glucose. It's glucose 1-phosphate that gets converted to glucose 6-phosphate. And the importance of that is that it can't actually take it all the way to glucose. And if it can't take it all the way to glucose, it can't ship it out because that phosphate group is going to kind of trap it in the cell. But your liver, it can take it all the way to glucose, and so it is going to ship out glucose to the rest of your body from all that glycogen it had stored up. So glycogen, awesome. What's not so cool is that there are a variety of metabolic disorders associated with mutations in the glycogen pathway. So there are mutations that affect things like glycogen synthase or the branching or debranching enzymes. These are associated with often fatal diseases such as Pompe disease, McArdle disease, various other diseases that can cause liver and kidney failure. They can cause muscle problems, various disorders caused by problems in this pathway.
So while we're on the topic of glucosal transferases, let's talk about how these different monosaccharides can actually get linked up and why this can matter. One example is your ABO blood type. So whether you're type A, type E, type AB, type O, This is dictated by which glycosyl transferases you have, so which sugar adders you have. And if you have different sugar adders, you add different sugars in different ways, and then this makes you have different sugar chains. And why this matters is that these sugar chains are going to get stuck onto proteins and lipids, and they can serve as antigens or things that bind antibodies. And so if you have a person who has a certain blood type, they're going to have that certain sugar. If you have that certain sugar, then your immune system is going to know that sugar is yours, and it's not going to try to attack it. But if you then give that person blood from someone else who had a different sugar, well, then this person, the immune system of the first person isn't going to know that that is okay. It's going to think it's foreign because, well, it is foreign, but it's going to attack it. And this can cause this blood type incompatibility. But at the heart of all of this is that people have different versions of this glycosyl transferase that then make it so that they're making different sugars. So the A protein is going to add an N-acetyl D-galactosamine. The B protein is going to add a different sugar, so it's going to add galactose. And the O version is inactive, and so people with this OO genotype only have this core chain. The plus minus is for a totally different thing. And then another thing is that some people actually have are secretors, and some people are non-secretors. And this has to do with whether these sugars are just on the surface, whether they just attach to lipid membranes of cells and just stick off of the cells, or whether they actually get attached to proteins that get secreted out of the cells. So just a fun fact for your day. Okay. So now let's talk about, since we just mentioned attaching these to things, let's talk about the different ways we can attach sugars to things. So remember that when we have sugar attached to another molecule, we call it a glycoconjugate. So conjugate is this word that we use when we describe two things stuck together through covalent bonds, so through the strong bonds. Okay, so let's start with glycoproteins. So as I mentioned, those sugar chains from those ABO blood types can actually get attached to proteins. And these can include proteins that are in the membrane. It can also get attached to membranes directly, but then we want to call it a glycoprotein. And then sometimes these proteins can get secreted out. And so we can have these on secreted proteins. We can have sugars on proteins that are membrane embedded, various things. We call it a glycoprotein when the protein is the main thing, and then you have sugar chains attached, and you can have different amounts of glycosylation. So some proteins might have just a couple chains, some might have a ton of chains, and then all those chains can have a ton of sugars, a few sugars. So there's all this variability, and you can even talk about sugar codes and things. There's a lot of variability in what that chain actually is, but there's only a couple places where it actually gets linked. And so we can talk about N-linked or O-linked. So N is it's getting linked through a nitrogen. And conveniently, it's linked through an amino acid whose abbreviation is also N. So asparagine has this nitrogen here that can then get hooked on to the sugar and the rest of the glycan. So this N glycans are getting attached to asparagine residues through the nitrogen. O-linked are going to link to an oxygen. And this oxygen, well, now we have two options where we can find this oxygen, either in serine or in threonine, where they both have this hydroxyl group that can then serve as a linkage point. And so then when you have a linkage through them, or you can also have this linkage through a hydroxylated lysine or protein. So lysine and proline can get modified. Lysine and proline, so you can see that they don't have a hydroxyl group as is, but they can get hydroxylated, and then they have an OH group that can serve as a linkage point. There's also differences between the N-linked and the O-linked in terms of how they get made. So with the O-link, you can add one and then add more. So with the N-linkage, you have this same big precursor thing added to all of the N-glycosylated sites. So it's not like you just add a single sugar. And so instead you add this whole thing, and then what happens is with bonsai tree, it gets whittled down and but you can also like add things to it. So it's not quite like a bonsai tree, but you can let it grow different ways, I guess. But basically, they have this core part stick around, and then this part can vary. So you can have there's different three main classes. So there's like high, oligomannose, hybrid, and complex. So yeah, it can get complex, and I'm not going to go into it, but you can see these different shapes are representing the various different sugars. And another thing is that you might think, okay, well, why do you add this whole big thing if you're just going to take off some of it? So even in the cases where you have complex things, you're still often modifying this top part or taking parts off. So you might think this is wasteful, but what happens is that the sugars, these proteins are often getting made and modified in various compartments inside the cell, like the endoplasmic reticulum and the Golgi body. So basically you have these compartments in your cell where this processing is taking place. And the protein is moving kind of through subcompartments in these subcompartments of the cell. And as it does so, there's different enzymes. So there's different like glycosyltransferases in different parts. And so when it's moving through, it's going to get modified in each of those parts if it's properly folded. And if it's not properly folded, then this can serve as a signal to target it to the ubiquitin proteasome system. So basically, it can target it so that this protein is then going to get degraded. Okay, so yeah, so it is useful. Okay, so that was a glycoprotein, where protein was the star, glyco was the minor role. Okay, now let's talk about proteoglycans. So here you have mostly sugar, and then part of protein, and the protein is often serving to help hold it in place. So some examples include heparin sulfate, chondroitin sulfate, and keratin sulfate, as well as mucus. So the sugars in proteoglycans are often glycosaminoglycans, or GAGs, and these are large polysaccharides, and they have a lot of modifications. These can include things like amino sugars, oxidation to carboxylate, and addition of sulfate groups. And these modifications, different proteoglycans can have different modifications, and these can allow it to serve as a sort of code that provides binding sites to proteins in the extracellular matrix. So the ECM, and it's going to allow them to act as scaffolds. So these are typically on these proteins that are like embedded in membranes and things, and they're going to then have these different sites where proteins can bind. This can alter the function of the protein. It can bring proteins together, all sorts of cool stuff. And those sulfate groups are, because they're negatively charged, they're going to like help soak up water. They're going to attract salt ions, so they're going to attract like sodium ions, and then the water is going to come with those, and you're going to get this to form this gel like we talked about before. And so this allows it to act as shock absorbers and lubricants and various things like that. So if you're wondering, mucus, so this is protein mucin, which is actually like a bunch of different proteins. But then there's our proteoglycans, so they have a ton of sugars on them, and they are allowing them to kind of make this jelly lubricatey stuff. Okay, next up, let's talk about peptidoglycans. So here we have sugar chains that are attached through short peptide linkers. And the classic place where you see peptidoglycans is in bacteria cell walls, in this periplasmic space between these membranes. These peptide linkers are going to like certify it, and specific linkers, the specific peptide parts, depend on the different bacteria. So this is Staph aureus, and you can see it has these glycan parts, but other bacteria will have different glycans. This is also a target of some antibiotics. These beta-lactam antibiotics like penicillin and ampicillin, they target the cell wall synthesis. Cells can get resistance, and this is how in the lab, we often take advantage of this by putting in this resistance gene, where basically it inactivates the antibiotic so that the walls can still get made. So that's just a cool little side note, or I guess not cool in terms of antibiotic resistance in the wild, but in the lab, it's very helpful and allows for us to select for things and stuff. Much more in other posts. Okay, so another group that bacteria have of these glycoconjugates that's really important are glycolipids. So lipids like your like membranes of cells. And then glyco, that's the sugar part. You might have heard of endotoxin. So that's LPS, lipopolysaccharides. And this is going to be a glycolipid that bacteria have, and different bacteria will have different ones, but these stick out from the outer membrane. You'll find these in gram-negative bacteria, and in order to help protect them, they have these lipopolysaccharides, which kind of stick out and make it harder to get to the bacteria's surface. It's called endotoxin, and it provokes an immune response. Glycolipids as well, lots of different types of glycolipids. Glycosyl phosphatidylinositol, so GPI. These serve to anchor non-membrane spanning proteins to the outside of the cell. So some membrane proteins are like go through the membrane, and some proteins can be actually just like anchored on the outside through this GPI. So you have one end of the sugar that's linked to a phospholipid head, and then the other is linked to the protein. So these phospholipids heads are these. So in the phospholipids surrounding our cells, they have this head group that is hydrophilic, so water likes it. And then these tails that are hydrophobic, so water excludes it. And then you end up with like a sandwich with the watery parts on either side. On this head group on the exterior, you have different head groups, and these can get modified in different ways, and they can get attached to proteins. And this is the link, a sort of linkage that is very helpful. And so here you have the protein, you have the sugar, and you have the lipid all acting together. So pretty cool teamwork. There's also things called cerebrosides and gangliosides, and these play important roles in the nervous system, and problems with processing them and making them can cause various medical conditions. Carolyn Bertozzi is a very well-known, like pioneering glycobiologist. So I've heard thank for a lot of what we know about glycobiology.